Welcome to The Explainer. Today, we're diving into a genuinely fascinating topic. What actually happens when the body's highly advanced defense network just breaks down? We're going to explore the incredible and sometimes chaotic world of immunology, taking a close look at how our internal safeguards operate and, more importantly, how they fail.
So when does the body's defense become its own worst enemy? It's a provocative question, right? We usually think of our immune system as our ultimate protector, like this relentless army fighting off foreign invaders, such as viruses and bacteria. But what happens when that army turns its weapons inward? We're looking at a complex internal system failure here, a cellular civil war where the very mechanisms designed to keep us alive actually begin to attack our own tissues.
To really understand this, we need to define exactly what we're looking at. Autoimmune diseases, or AD, are pathological conditions where an aberrant or abnormal response of the innate and adaptive immune systems leads to a loss of tolerance to self, attacking the body's own tissues. Throughout this explainer, we're basically going to act as detectives, investigating the suspects, the environmental crime scenes, and the broken cellular failsafes that lead to this loss of tolerance.
We've got a lot of ground to cover, so here's a quick look at the agenda. 1. Normal immunity versus autoimmunity. 2. Genetic susceptibility and risk. 3. Environmental triggers and lifestyle. 4. Cellular mechanisms of autoimmunity. 5. Cell clearance and death. And 6. The clinical disease spectrum.
Okay, starting with Section 1, normal immunity versus autoimmunity.
Now, what's really interesting here is the stark contrast between healthy function and disease. We actually have a baseline level of self-recognition that is perfectly normal. It's not a flaw, it's a feature. Under physiologic autoimmunity, our body produces IgM antibodies. These are unmutated, they have a moderate affinity to our own antigens, and they essentially act as natural housekeeping, a first line of defense, and a cleanup crew. But in pathological autoimmunity, the system goes rogue. The immune system starts turning out somatically mutated, high-affinity IgG autoantibodies. These are highly targeted, destructive weapons that disrupt cell clearance and cause severe tissue damage.
That brings us to Section 2, genetic susceptibility and risk. So how do we cross that line from healthy housekeeping to pathological attacks? We have to look at the Swiss cheese model of risk. Genetic mutations alone aren't usually enough to cause disease, but they lay the blueprint for susceptibility. Look at the suspects in this lineup. The HLA-DRB1 gene, specifically the shared epitope, is heavily linked to rheumatoid arthritis because it aberrantly presents modified peptides. But then look at non-HLA alleles like STAT4 and PTPN22. These disrupt interferon production and the selection of T and B cells. This brilliantly illustrates how single genetic flaws can overlap across multiple diseases like RA, SLE, and type 1 diabetes. We even see rare monogenetic mutations like AIRE, which single-handedly impairs central tolerance. The takeaway here is that a variety of overlapping genetic flaws set the stage for these diseases.
Next up, Section 3, environmental triggers and lifestyle.
Like we said, genetics are just the first layer of the Swiss cheese. The second hit comes from external environmental factors that trigger those genetic landmines. Cigarette smoking is a huge one. It physically alters biology by promoting the citrullination of proteins in the lungs, basically changing their structure so the immune system doesn't recognize them as self anymore. Then you've got physical factors like silica and solvents. Dietary lifestyle factors play a massive role too, especially low vitamin D, low omega-3s, and highly processed sugars. Throw in gastrointestinal microbiome dysbiosis, ultraviolet light, and specific medications, and you have a perfect storm of triggers stressing the immune system.
To put this into perspective, just having the HLA-DRB1 shared epitope increases your risk for rheumatoid arthritis by 4 to 6-fold. But if you combine that genetic blueprint with the environmental trigger of smoking, your risk catapults to 20 to 40 times higher. It's an absolutely massive amplification of risk.
Moving right along to Section 4, cellular mechanisms of autoimmunity.
Let's see how all this builds into actual cellular chaos. One of the most fascinating mechanisms here is molecular mimicry. Imagine you get a simple throat infection with group A streptococcus. Your body creates antibodies targeted right at the strep M protein. But here's the fatal flaw. That bacterial protein looks structurally very similar to the myosin proteins in your own heart. Confused by the resemblance, your antibodies cross-react. They deposit in the heart, causing severe inflammation, which leads to rheumatic fever. The pathogen literally tricked the immune system into friendly fire.
You know, the immune system is basically this massive, radiating, interconnected network.
It relies on a super delicate balance between T cells, B cells, and antigen-presenting cells. But in autoimmunity, we see the rise of age-associated B cells, or ABCs, alongside refractory long-lived plasma cells. When tolerance breaks down, these rogue players form a vicious, self-perpetuating cycle, continuously pumping out high-affinity autoantibodies.
And these cellular autoantibodies translate directly to clinical reality for the patient. Depending on what the rogue antibody targets, you get a highly specific, devastating disease. If antibodies target type 4 collagen, you get Goodpasture's disease, destroying the kidneys and lungs. If they target citrulated proteins, you get rheumatoid arthritis, leading to bone damage. If they target double-stranded DNA, you get systemic lupus erythematosus, causing system-wide tissue damage.
Which brings us to Section 5, Cell Clearance and Death.
This is the final failsafe in our system. Because even if the immune system creates rogue antibodies, it still needs a target. So what happens when the body's garbage disposal breaks down? Well, we have to look at the types of cell death. Apoptosis is programmed cell death. It's controlled, clean, and crucially, it does not induce inordinate inflammation. But then you have necrosis, unprogrammed cell death, alongside highly inflammatory pathways like netosis, pyroptosis, and necroptosis. These are messy. The cell bursts open, spilling its contents into the tissue, acting as a massive danger signal that absolutely drives severe inflammatory responses.
Normally, a healthy body sweeps up this debris, but with defective clearance, say a genetic mutation in an enzyme like DNAC1, your body's garbage disposal completely fails. It can't clear out extracellular debris like neutrophil extracellular traps or nets. This leaves exposed DNA and cellular contents just sitting there in the tissue. This uncleared debris is the exact fuel autoreactive B cells need to create pathogenic immune complexes, driving the widespread systemic inflammation we see in lupus.
Finally, Section 6, the clinical disease spectrum.
Let's pull this all together. When we combine genetic susceptibility, environmental triggers, broken cellular tolerance, and defective clearance, we arrive at the clinical outcome. And it exists on a vast spectrum. On one end, we have highly targeted, organ-specific attacks. Graves' disease takes laser-focused aim at the TSH receptor in the thyroid. Type 1 diabetes is directed entirely at beta cell antigens in the pancreas. On the other end of the spectrum, we have massive systemic wars. Lupus targets dsDNA and ribonucleoproteins, meaning the attack can happen anywhere in the body. Rheumatoid arthritis targets citrulinated peptides, devastating joints while causing systemic inflammation.
So we've mapped the crime scene. We know the genetic suspects, the environmental triggers, and the cellular weapons, which leaves us with this final thrilling question. If we can map the exact genetic and cellular failsafes that cause these system failures, can we learn to proactively target them? Could the future of immunology move beyond just treating symptoms and actually repair the failsafes to cure autoimmune diseases? It's an incredible frontier of medicine, and the deeper we look into our own cells, the closer we get to the answer.